Okay.
Today we'll be starting off with your AS Biology chapter number 4, which is cell membranes and transport. Can you tell me what is the main function of cell membrane in eukaryotes and prokaryotes?
It is to allow the substances from the environment uh, to, uh, to enter the cell, basically.
Okay. Good. It can control the movement. It can control what comes in, what leaves
Yeah.
the cell. It can also control your diffusion, osmosis, active transport. Everything happens through its membrane. It can provide you with protection. It can prevent large molecules from entering. It can also keep a little bit of the shape. It is also acting as a boundary for all organelles. It is a layer that is separating the intracellular structures and the extracellular structures. एक बीच में layer separate कर रही है. It is also very important क्योंकि ये animal cells की outer most layer होती है. Animal cells में cell membrane के बाहर तो कुछ भी नहीं, it's the most outermost layer. So in this chapter, we are going to describe the structure of the membrane, which is made up of phospholipids, and हम इसका एक fluid mosaic model से explain करेंगे of a membrane structure. Then we are going to describe the arrangement of molecules in cell surface membrane and the roles of the molecules found in cell surface membrane. This is going to be our today's agenda. कल हम करेंगे outline the process of cell signaling and how substances enter and leave the cell. ये हमारा होगा टुमारो एजेंडा. और वीकेंड पे अगर हमने एक एक्स्ट्रा क्लास मैंने रखी, आई विल सी इफ आई एम अवेलेबल, तो हम ये थर्ड 3 पार्ट्स के अंदर वी आर गोइंग टू फिनिश दिस चैप्टर बाय दिस वीक. So what is the importance of membranes? In chapter 1, you saw that all living cells are surrounded by a very thin membrane, which is known as the cell surface membrane. This controls the exchange of materials such as nutrients and waste products between the cell and its environment. Inside the cell, it acts as a regulation of transport across the membranes of organelles, which is also vital. And it has other important functions as well. For example, receiving hormone messages. अब this is something new that you'll see कि यहाँ पे इसका cell signaling में भी role है. हार्मोनल मैसेजेस, आपका हार्मोनल कम्युनिकेशन होती हैं, सिग्नल्स आ रहे हैं, अफेक्ट आपने जनरेट करने, यू हैव टू जनरेट अ रिस्पॉन्स, होम्योस्टैटिक रिस्पॉन्सेज हैं. प्लस क्लोरोप्लास्ट के अंदर भी आपके पास जो थिलाकॉइड मेम्ब्रेन सैक्स होते हैं, जो क्लोरोप्लास्ट के अंदर भी मेम्ब्रेन्स होती हैं इस तरह करके, ये भी मेम्ब्रेन्स हैं. वी कॉल दीज द थिलाकॉइड मेम्ब्रेन्स. और ये थिलाकॉइड मेम्ब्रेन्स के ऊपर आपके पास लगा होता है क्लोरोफिल, विच आर एक्चुअली रिस्पॉन्सिबल फॉर एब्जॉर्बिंग sunlight, which you have already seen in photosynthesis as well. It is also important to study the structure of membranes to understand how these functions are actually achieved. तो अब हम स्ट्रक्चर मेम्ब्रेन्स पढ़ेंगे, विच इज समथिंग वेरी न्यू जो आप एएस बायो में पढ़ते हो. फॉस्फोलिपिड्स, नाम से पता लग रहा है. फॉस्फोलिपिड्स में है जी फॉस्फेट है और साथ यू हैव लिपिड. और लिपिड्स की बात की जाए तो वी आर गोइंग टू डिस्कस द डिफरेंट फैटी एसिड चेन्स दैट आर एक्चुअली a part of the membrane structure. Understanding the structure of membrane depends on the understanding the structure of phospholipids. Phospholipid की एक बायलेयर बनेगी. बायलेयर का क्या मतलब है? पहली लेयर और साथ ही दूसरी लेयर. दिस इज नोन एज अ बायलेयर. बाय मतलब टू. Figure 4.2 shows what happens in the phospholipid molecules spread out over a surface of water. This is what a phospholipid molecule looks like. एक phospholipid molecule के दो parts होते हैं. The head is made up of phosphate and the tails are made up of lipid. अब जो ये लिपिड टेल्स होती हैं, दे हेट वाटर, बट द फॉस्फेट हेड्स दे लव वाटर. वी कॉल दीज हाइड्रोफिलिक पोलर हेड्स. हाइड्रो मतलब पानी, फिलिक मतलब लविंग, वाटर लविंग हाइड्रोफिलिक पोलर हेड्स हैं, विच आर मेड अप ऑफ व्हाट? फॉस्फेट. और जो हाइड्रोफोबिक नॉन पोलर टेल्स है, वाटर हेटिंग, ये बनी हुई है आपकी लिपिड से. सो यू कैन सी कि जितने पोलर हेड है वो कहाँ पे पॉइंट करें? टुवर्ड्स द वाटर. अगर आप उनको सिर्फ एक सरफेस में भी देखोगे, पानी की सरफेस में, तो आप देखोगे वो एक ऐसा स्ट्रक्चर बनाएंगे कि सारे फॉस्फेट हेड्स ऑटोमेटिकली दे आर ऑल पॉइंटिंग आउट, because the phosphate heads are water loving and the hydrophobic lipid tails are actually water hating. You can see over here in this structure as well, all those hydrophilic phosphate heads are actually pointing outwards. और membrane का structure ये है. This is what we call a phospholipid bilayer structure of a membrane, जो आपको draw करने को भी आएगा. You have to draw this in the paper as well. This is the one that you have to draw. So the heads are hydrophilic. Their tails project out of the water because they are non-polar hydrophobic. Polar refers to an uneven distribution of charge. Polar means charged. Polar means charged. And water is also polar. If water is also polar and phosphate molecules are also polar, that is why they are loving each other. If the phospholipids are mixed with water, they form either ball-like structures or sheet-like structures known as bilayers. We have to focus on the bilayer structure because this is what forms the membranes of your cells. In a mycel structure, all the hydrophilic heads face outwards into the water and they shield the hydrophobic tails from the water. In the middle of the ball, the tails are actually pointing towards each other, creating a hydrophobic environment inside the mycel, as shown in figure 4.2. In bilayers, the hydrophobic tails are also shielded from the water by hydrophilic heads, like you can see in all of these diagrams. So it is now known that the phospholipid bilayer is the basic structure of the membrane. और bilayer का मतलब है two layers as shown in all of the figures above. One very important thing is the width of membrane. The width is what? 7 nanometer wide. 7 nanometer का क्या मतलब है? 7 into 10 to power minus 9 meters is the width of these membranes. Now membranes also contain proteins. And these proteins can be seen in the electromicrographs as well. And due to the presence of those proteins, we call the structure of membrane as a fluid mosaic model. Fluid क्यों कहते हैं? Because we have these hydrophilic heads, but in between we have some protein molecules as well, like this. फिर एक और protein molecule आ जाएगा. Then we have again this structure. Then same is the case here. This is how a bilayer is formed. But in between, you can see that there are what? Some proteins as well. We have small proteins हैं, कुछ channel proteins भी होती हैं. And in between these phospholipids, we also have some lipid molecule as well. Cholesterol molecule भी हम इसको कहते हैं. We call these the cholesterol molecules, which can actually help us in more fluidity. So this is what makes up the bilayer structure. But why do we call it a fluid mosaic model? Can you tell me what is the meaning of fluid? मेम्ब्रेन स्ट्रक्चर को फ्लूइड क्यों कह रहे हैं? स्पीक
It is
लाउडर.
because that cholesterol provides helps the shape to like split up, kind of like slip down.
यस, गुड है. वो एक फ्लूइडिटी प्रोवाइड करती है. और मोज़ैक का क्या
Yeah,
मतलब होता है?
like
एब्स्ट्रैक्ट का नाम सुना हुआ है? जैसे वॉलपेपर्स होते हैं फोन के,
yeah,
एब्स्ट्रैक्ट. एब्स्ट्रैक्ट वॉलपेपर क्या होते हैं? कुछ भी नहीं है, लाइनें मारी हुई है, वॉलपेपर बन गया. ये क्या है? कुछ भी नहीं है, सिंपल एक पैटर्न है.
yeah.
एक इरेगुलर पैटर्न है. ठीक है, मोबाइल की वॉलपेपर डाउनलोड करो, यू हैव अ मोज़ैक ऑप्शन. क्या है? नथिंग. इट्स जस्ट अ रैंडम आर्ट. सो ये जो डिफरेंट रैंडम आर्ट बनते हैं, अ मिक्सचर ऑफ कोलेस्ट्रॉल, प्रोटीन, फॉस्फोलिपिड, दे कॉल फॉर अ मोज़ैक मॉडल, which is like a variety of structures present है एक ही molecule के अंदर. In 1972, two scientists, Singer and Nicholson, used all the available evidence to put forward a hypothesis for the membrane structure, and they called their model as the mosaic model. It is described as fluid because both the phospholipids and the proteins can move about by diffusion, and the phospholipid bilayer has the sort of fluidity you associate with olive oil as well. The phospholipid molecules move sideways in their layers, and some of the protein molecules also move about within the phospholipid bilayer, like the icebergs in the sea. Others remain fixed to structures inside or outside the cell, and the word mosaic describes the pattern that is actually produced by the scattered protein molecules. तो आपको एक क्वेश्चन आ सकता है, and you have to copy down all of these because this is the best answer that is already written for you in the book. Book has the best answers for all your past papers question. Remember that. So mosaic क्यों है? Because of the scattered protein molecules within the surface of the membrane. So fluid mosaic model is the currently accepted model of membrane structure proposed by Singer and Nicholson in 1972, in which protein molecules are free to move about in a fluid bilayer of phospholipid molecules. So these are the different features of fluid mosaic model. We first know that it's a double bilayer structure made up of phospholipid molecules, and they all keep moving around by diffusion. मतलब कि these phospholipid molecules are fluid, they can move around the membrane as well. They can change the directions, but they should be within the structure of the membrane. बाहर नहीं, मतलब अंदर अंदर move करते रहो. The phospholipid tails point inwards, facing each other and forming a non-polar hydrophobic interior, water-hating interior. सारी tails जो हैं, you can see all of the tails are pointing inside, but the hydrophilic heads, they are all pointing outside in the cytoplasm or outside in the blood or the tissue fluid. The phospholipid heads faced outwards into the aqueous water containing medium that surrounds the membranes. Some of the phospholipid tails are saturated and some are unsaturated. Saturated क्या होते हैं? Carbon single bond carbon. And unsaturated means carbon double bond carbon. The more unsaturated they are, the more fluid the membrane is. This is because the unsaturated fatty acid tails are bent and therefore they fit together more loosely. Fluidity is also affected by the length. Remember, longer the length, less is the fluidity. And as the temperature decreases, membranes become less fluid. But when you increase temperature, fluidity increases. But some organisms which cannot regulate their own temperature, such as bacteria and yeast, they respond by increasing the proportion of unsaturated fatty acids. Now, in many organisms which are not affected by temperature, even if you increase the temperature, the fluidity doesn't increase. So that is why they convert their saturated phospholipids into unsaturated fatty acids in their membrane. So that more the unsaturated fatty acids, more would be the fluidity. Proteins may also be found in the inner layer, and for that particular reason, the presence of proteins called this the mosaic model, क्योंकि those proteins were randomly present, small हैं, large हैं, and the outer layer most commonly spanning the whole membrane, they are known as transmembrane protein. कुछ membranes होती हैं that are like this, all across the membrane, मतलब यहाँ पे पूरा they have formed. We call these transmembrane proteins. फिर कुछ आती है like these intrinsic proteins. Some are pointing outwards, extrinsic proteins. And the proteins also have a hydrophobic and hydrophilic polar regions. Hydrophobic would be pointing inwards towards the membrane, and hydrophilic would be pointing outwards. They stay in the membrane because the hydrophobic regions made from hydrophobic amino acids are next to the hydrophobic fatty acid chains, which in the watery environment either side of the membrane. The hydrophilic regions made from hydrophilic amino acids are repelled by the hydrophobic interior of the membrane, and therefore they face into the outer. So this is an amazing diagram that you are going to draw right now at the end of the class as well. And you have to make the exact diagram. I want to see the proteins. I want to see glycoproteins. I want to see cholesterol molecule, the phospholipid, label the hydrophilic head, label the hydrophobic tail. You have to make these glycolipids, and you have to make these random glycoproteins. Here you have to make this transmembrane protein as well. And transmembrane protein can be either channel proteins or they can be carrier proteins as well. Because mostly these transmembrane proteins are acting as transport proteins as well. So this is what you have to make at the end of the class. This is a good diagram. Okay, this is also a good one. That is also a good one. Okay, this is recommended. It's more detailed, and you should have to be drawing this as a proper label diagram. Use different colors, okay, for a label diagram, and then send it to me at the end of the class. So most of the protein molecules float like icebergs in the phospholipid layers, although some are fixed to the structures inside or outside the cells, and some do not move about. Many proteins and lipids have short branching carbohydrate chains forming glycoproteins and glycolipids. They have a very important role in your cell signaling. अगर आपके पास glucose molecule bind with a lipid molecule, they form glycolipids. And if this glucose molecule binds with any amino acid or protein, they form glycoproteins. Glycolipid, glycoprotein. दोनों membrane के ऊपर as chains attached होते हैं, like this. Molecules of cholesterol are also found in the membrane, and we are going to study that एक function का what is the role of each of the functions and role of the each molecule that are actually found in the membrane in the next topic.